Okay, so I've had a lot of questions about transcription and translation, how you might draw it on your poster. So I'm just going to draw a quick version of it here and to give you an idea of how I might do this. So it looks a lot like DNA replication, so we're going to start with a picture like that where we have our DNA strand that's been opened up, right, so that we can get at the gene that's in there. Okay, so this top strand is going 5' to 3', bottom strand is going 3' to 5'. Okay, it's our DNA, whatever. All right, and let's just go ahead and put some letters in here. I'm just making it up here. Okay, so if that's the top strand, then the bottom strand is going to be complementary. So if I have an A, I've got a T. A, C, G, T, G, C, G, G, A, C, C, and so on. Okay, and let's say that this top strand, because we read DNA 5' to 3', that means that this top strand, if we're going to read it left to right, the top strand will have the gene. Okay, so let's say that this is the gene right here. Okay, which means that this top strand is the coding strand, which means that the bottom strand is the template strand. Okay, and this is all DNA. Now obviously we would actually start transcribing this gene way back here at the TATA box, wherever that might be. Okay, and we'll end over here at the polyadenylation signal. Okay, so but that's our gene right here. This is where our exons and introns and all of that are going to be. And so, because this is RNA and being made out of RNA, we are going to have our RNA polymerase is going to do it, and we don't need a primer. So the enzyme is going to come and sit down, and it's actually going to recognize this TATA box, or ATA, because that's what it'll be on this side, and it's going to sit down on the DNA strand. This is our RNA polymerase. And it's going to start copying, building an mRNA in the 5' to 3' direction. Okay, so it's going to read it 3 to 5, but it's building the new strand 5 to 3. And so it's going to see this T, it's going to write an A, it's going to see the A, it's going to write a U because T's are not used in RNA. C to G, G to C, T to A, G to C, C to G, C, C, U, G, G, etc. Okay? And this right here is your messenger RNA. That's your mRNA. And you'll notice that the mRNA and the code are identical, right? Except for that the mRNA has U's in place of the T's. And if that confuses you, I would just write it complementary just like this and then erase all my T's and put U's. Anyway, so then this is eventually, once it's finished, it's going to come out. Here's our mRNA, okay, and it starts, we'll just put the starting letters on there, okay. This is our mRNA, and it's running 5' to 3', okay, mRNA, we'll label that. All right, so now there's a couple modifications that have to be done. There's 3 modifications that have to be done before it leaves the nucleus. Number 1, we have to get rid of the junk sequences, and I haven't marked these here, but on a test or something they would be marked. But like, let's say from here to here is an intron, and then these 2 are exons. So we need to get rid of this intron in there and splice the rest of it together. The other thing that happens is we put a modified guanine 5' cap on there, and then we add a bunch of A's, or a poly A tail, to the 3' end. So now this mRNA is ready to go out and be read by the ribosome. Okay, so we'll just move this up and we'll do a ribosome. So this, by the way, this whole process that we just drew here is transcription. Okay, think of a medical transcriptionist. We're just taking it from one version of it to another version of it. It's the same language. It's still written in nucleic acids, but we're transcribing it into a language that we can use and pull out of the nucleus. All right, so then that mRNA is going to come to a ribosome outside of the nucleus in the cytoplasm. So you can make, I don't know, you can make this be our nucleus. This is going to go out into the cytoplasm here. So here's our mRNA. We have our 5' cap, our mRNA, and our poly A tail. Okay, and we've got our amino acid, or our nucleotides, AUG. We'll just put a couple down, CAC, etc. Okay, so this is where the ribosome comes in. And the ribosome is going to sit down, and this is sort of hard to do in 3D and moving, but the ribosome is going to sit down. It actually has 2 pieces, and it has 3 sites. Okay. This is where we'll just EPA. All right, so when it sees, it'll recognize this AUG in the recognition site, and it'll bring in a tRNA. All right, we'll do that in black. So our tRNAs, or transfer RNAs, they bring an amino acid to the mRNA that matches this particular codon. So let's actually label this. This right here, AUG, that is the codon. And it looks just like the coding strand, right? It looks just like the coding strand of the DNA. Okay, and then the tRNA is going to come in and sit down right here. They're kind of curved, right? And they're going to bring an amino acid with them. Now this particular one will have an anti-codon right here that'll be complementary to this. So it's going to be UAC. But remember, when we read the genetic code, we read it in terms of the codon. So we'll go to the genetic code, that universal genetic code, and we'll see AUG, and we'll see that it codes for methionine. So that means that the UAC tRNA is going to be bringing a methionine. Okay, and then the ribosome will just click over to the next site. So this will end up over here, and then the next one, a tRNA will come in, okay, and it's CAC, so it's going to have GUG, and it's going to be bringing an amino acid, and I didn't look it up, I don't know which one it is. Okay, and that'll click over. So in the P site is where a peptide bond is made between these two amino acids. And then it'll click over again to the E site, and you'll get another one in here with another amino acid. It'll make another peptide bond. E stands for exit, so this empty tRNA that's just been used will go out and go get replenished with another methionine, so it can come in and do it again. So it just clicks along until it hits a stop codon. So somewhere at the end it's going to have a stop codon like UAG, and no tRNA, so there's not a tRNA that matches that, and so the ribosome will drop off, and you'll end up with this long polypeptide chain, okay, which is your protein. All right, so you could draw this in like 3 steps where the ribosome is continuously moving over, right? So I don't know if I could draw this, but in step 2, our 5' cap and our poly A tail, the AUG, CAC. The AUG is now in the P site of the ribosome. I'm not very good at drawing this. Okay, this is the P site. This is the A site. This is the E site, right? You've already got the methionine on there, and you've got the next amino acid. Wow, that was ugly. On there, okay? And then it's going to click over one more time to the E site. We'll just go ahead and draw that. Why not? So we'll move this up a little. Here's our last one. These are poly A's. Oops. Your AUG, CAC, and whatever. And at this point, the ribosome, the AUG is in the E site, P site, A site. And this particular transfer RNA is now empty, and it's going to exit. And then these two are still in here. The methionine's already attached. Wow, I am not good at drawing those. And you're just going to click on, click as you go along and get a polypeptide. And that is translation, because we're taking it from one language, nucleotides, into another language, which is amino acids or proteins. All right, so that's transcription and translation. That's what you should have on your poster and explain to people. All right.